Welcome again to today's Asciminib conversation featuring CML highlights of the recent EHA2026 Congress that took place in June in Stockholm in Sweden. So the idea of this webinar is this is for people who were not able to attend the Congress in person, or for those of you who'd like to have or get a comprehensive overview plus a put into perspective from our two experts. We're very privileged and delighted to have two international experts here with us here today that are probably very familiar to all of you, but I will definitely introduce both of them later into more detail. But please welcome Professor Andreas Hochhaus from Jena in Germany and Associate Professor Katerina Polakova from Prague in the Czech Republic. To give you a brief overview of the program today, so we have round about 45 minutes for the webinar. That's divided into two parts. We will start with clinical aspects from EHA presented by Andreas Hochhaus, then immediately proceed to the biological aspect that will be presented by Katerina Polakova to us. And then if there are not any burning questions in between, we reserve some time, around about 15 minutes, for a Q&A session with lots of time for your questions. But please feel free to submit your questions throughout the whole webinar and both of the presentation. Now, let's get started with the clinical part, and it's my pleasure to introduce Professor Andreas Hochhaus into more detail to you. He is Director of the Department of Hematology and Medical Oncology, Coordinator of the University Tumor Center, and Co-Director of the Comprehensive Cancer Center in Central Germany at the Jena University. He is also full professor for internal medicine, hematology, and oncology at the Friedrich Schiller University in Jena. As you may all know, he has played a leading role in international clinical trials of TKIs from Imatinib to Asciminib and future developments, and has contributed substantially to advances in treatment optimization, molecular monitoring, and treatment-free remission in CML. He is also co-chair of the German CML Alliance and is also focused on enhancing access for patients to clinical trial and driving active patient participation. Not to mention that he has published, I think nowadays, more than 700 peer-reviewed papers, and is editor-in-chief of the Leukemia Journal. So again, we're very privileged and delighted to have you here, and I stop sharing, and we're looking forward to your clinical highlights of the meeting.
Yeah, Stephanie, thank you very much for this kind introduction, and I'd like to share my screen here. This was enough from all this listing of CVs and so on. I think we should go into the details of what we have seen in Stockholm. Actually, this is the Stockholm's Messen, the fair in Stockholm. It was important to have such a big hall because more than 10,000 people attended this meeting. It was actually in parallel to the 50th wedding anniversary of the Queen and the King of Sweden, and this was very interesting to see this in parallel here during this EHA meeting. What is important in the clinic of CML? We celebrate this year 25 years of tyrosine kinase inhibitors. When we started with Imatinib, the main goal was, of course, survival and to avoid blast phase disease. Later, when we developed quantitative PCR, when we have seen that MMR is an important predictor for survival, then the molecular remission and later also deep molecular response and treatment-free remission became very important. And now in the 20s, now we more and more focus on side effects of the drugs, on tolerability, and in particular on patient-reported outcome. That means quality of life. And this background is important to understand the importance of Asciminib as BCR-ABL-specific inhibitor without the inhibition of alternative kinases And the Ask for First study was summarized in the CML session by Tim Hughes with the so-called 144-week analysis. Week is missing here. Week 144, yeah, analysis. It's a little strange, but in some pharma companies, for instance, Novartis, a year has 48 weeks, and therefore we have now the 3-year analysis, which is 144 weeks. And just briefly to introduce Asciminib with this myristoyl site, which is inhibited by this drug and which imitates the autoinhibition of ABL with this myristoyl tail. And Ask for First is very well known now with this third update. A randomized study for newly diagnosed CML patients comparing Asciminib with Imatinib, and in a different stratum, Asciminib with second generation tyrosine kinase inhibitors. MMR had been the primary efficacy parameter after 48 weeks, but now, as I mentioned, we have the 3-year analysis, and of course we look at deep molecular response as well. How many patients are still on drug? And here is a comparison. You see that there is a clear advantage for Asciminib, more than 80% in the Imatinib stratum, 76% in the second generation stratum. And the discontinuation was mainly based in the comparator arms on treatment failure, 18% Imatinib, 8.8% second generation, and of course also on adverse events. That means what was expected, that Asciminib as a BCR-ABL specific drug is better tolerated, is very clear here. Less patients discontinued due to adverse events. Main comparison, MMR on the right, 144 weeks, 79% Asciminib, 47 Imatinib. The comparison with second generation, 75 versus about 60% with second generation drugs. That means clear advantage for Asciminib. So, and now who should be treated with Asciminib? When we look at the comparison to Imatinib, I think it's almost everybody who has an advantage of Asciminib in this forest and plot analysis. Yeah, a very clear advantage actually. For second generation, there's a little overlap with the 0 line here, but overall, independent of Framingham, independent of age, of sex, and in particular on ELTS score, I think that's the most important parameter here. We have an advantage for Asciminib. Deep molecular response is the prerequisite for treatment-free remission. This is planned in the study after 5 years of treatment. Now we have 3 years. That means we have time to go. But we see on the right, MR 4.5, 43% versus 19 compared to Imatinib. That means a very clear prediction that more patients are eligible, will be eligible for treatment-free remission in this stratum, but also in the second generation, we have 41 versus 29%, and we have 2 years to go to improve these numbers and then to start treatment-free remission, which is of course not mandatory, but we advertise this for the patient. That's an option, and we ask the patients to do this. In parallel, we have the Ask for Start study, and in the Ask for Start study, we advertise for treatment-free remission after 3 years of treatment. That means when we compare these two studies, we will have a comparison of a 3-year discontinuation minimum versus 5 years, which is of course very nice to have this, because we expect that everything goes faster with Asciminib, and it's a question whether 3 years are enough or 5 years. But here in this study, we plan for 5 years of treatment. We have, of course, as with all monotherapies, we see mutations very early in the course of disease. In the course of the disease, we had 10 patients with Asciminib-induced mutations. These are mutations of the myristoyl site, and also in all the other cohorts, we had mutations. But we have the feeling that after achievement of a deep molecular response, we don't see mutations anymore. In the third year of therapy, we had no mutations in the Asciminib arm, but 2 mutations in the Imatinib and Nilotinib arms. That means it might be a problem of the time when molecular response has not been achieved and mutations are selected. That means we expect a better, more stringent treatment over time without mutations in case of deep molecular response with Asciminib. So now tolerability. Most important are the adverse events leading to discontinuation. This was the case in 6% of Asciminib patients, 13% Imatinib, 13.7% with second generation drugs. That means much better tolerated. And these are here now the details. For Imatinib, for instance, the most burning problem are the myalgias, which happen every day in some patients. We have seen this in 18% and muscle cramps in 21%. When we look at the muscle cramps for Asciminib, it's only 4% of patients. That means it's a very clear reduction. We see cytopenias with Asciminib as well compared to Imatinib. That's part of the efficacy of the drug and the slower recovery of normal hematopoiesis. That's a transient phenomenon. Actually, we asked Novartis to do an analysis by timing. That means to see which side effects happen in which half year of therapy. This will be shown at ASH, not in this analysis here. So, and for second generation, we have mainly the liver tox for Nilotinib and Bosutinib, which is not the case with Asciminib. In discussion are the arterial occlusive events. When we look at 100 treatment years, patient treatment years, we see it now in 1.3% of patients compared to 0.5 with Imatinib and 1.1 with second generation. That's very low numbers. Looking at considering the patient age range here, but it's a signal. That means we need to observe this on Asciminib as well. We see more hypertensions with Asciminib as well. That means there is a signal for cardiovascular disease, but this needs to be observed in the next years of therapy. When we summarize this, and this is from Jorge Cortez from ASH, when we summarize this, when we see a newly diagnosed CML patient, we should ask him for his treatment goals. Is it survival? Is it treatment-free remission? Young family, wants to have children, and so on. And over time, it will be more and more tolerability. And when we consider these parameters, we have different scenarios. For survival, we can start with any drug because we don't see survival differences. But when we look at treatment-free remission, it's very likely that Asciminib will be dominating because of the rates of deep molecular response. And also for tolerability, patient-reported outcome is a clear advantage for Asciminib. Arterial occlusive events, we don't know. Imatinib is definitely the best, but if Asciminib has an advantage over second-generation drugs, is still open and needs to be discovered in the next years. ELTS risk plays a role, of course, for overall outcome, for the chance to achieve deep molecular response, but there is a clear advantage for Asciminib as well. We have a point with A6L1 mutation. This was not discussed here in this talk, but patients with A6L1 mutations have a slower response rate, a lower response rate with Asciminib, which is not disadvantageous compared to second-generation drugs, but of course compared to Asciminib-treated patients without A6L1 mutation. That's an open point. This needs to be discussed with new studies, for instance, with combination treatment studies. The second talk in this session was presented by Dennis Kim, the ENABLE study. That's based on a new compound, an ATP competing drug, ELVN-001, which has several advantages over the current ATP competing drugs. It has a different pharmacokinetic profile. It can be easily combined with other drugs because it has no cytochrome 3A4 activity, and is therefore a very good combination partner, which I already mentioned, and we will see how this will go on when this phase 1 here is completed. You see here how specific it is. It's also a very specific ABL inhibitor compared to all the other ABL inhibitors on this slide. And Dennis reported on this phase 1 study, phase 1A, with typical increase of the dosages in different cohorts, and then phase 1B with the expansion cohort 60, 80, and 120 milligrams. The optimal biological dose was defined when we compared efficacy with tolerability to 80 milligram ELVN-001 once daily. Side effects were presented on the 80 milligram cohort. Specifically, there were some cytopenias in late stage disease, also some lipase increases without pancreatitis. That means overall it was very well tolerated. That's my personal experience as well. And specifically, very low incidence of arterial occlusive events. Here are the MMR rates for all patients, then for all patients from the phase 1B with the efficacious dosages, and specifically in orange, the 80 milligram once daily cohort, and we see an MMR rate of about 50% in the cumulative response analysis, which is, I think, very good in the late stage disease. And here on this chessboard, you see how the responses, the molecular responses, improved over time. Anything which is in white here, in white color numbers, show an improvement. The black numbers show a stable molecular response, but there was no patient who had a reduction of the molecular response over time. So, and now after Asciminib, it's of course of interest how ELVN-001 behaves after Asciminib failure. And these data are also very good. Of course, we have different lines of therapy, and post-Asciminib on the right, patient who had only 1 to 2 lines, and one of them was Asciminib, had an achievement of MMR of 60%, which is, I think, again, a very good point. That means we need to now to discuss and to develop a phase 3 study for the registration of ELVN-001. This will be the next step, and this is ongoing. The study will start at the end of this year or early next year. The next point is, and I've seen Meinolf Suttorp here in the audience. Hello, Meinolf. The Ask for Kids study. Ask for Kids 1 was not called 1, but just the Ask for Kids, tested Asciminib in the usual formulation. But there was an advertisement on a poster for the Ask for Kid 2 study, which I think is very relevant and very important, because Novartis will introduce a new formulation of Asciminib, which can be taken after a meal. And this might be very important for kids. And it's, of course, also important to have a dose range for the kids, just to adjust the dose, which is not possible with tablets, with a formulation which can be dosed much more specific, much more accurate for kids. And this will be tested in this Ask for Kid 2 study. So that's the important background for kids. Has been published by Markus Metzler in Lancet. A twin pair, the smaller one had CML and was treated with Imatinib and Dasatinib, did not grow very well compared to his twin brother. and after switch to Asciminib, he matched up with the growth, and I think that's the clear advantage of Asciminib for kids, that the growth is not inhibited with Asciminib compared to all the ATP competing drugs. My last study, which I want to introduce, is the NAUT study. This was presented by Susanne Sausele, a German study for treatment-free remission after one failure, at least one failure of treatment-free remission, a second stop study. And this is the study design here. The patients were on any TKI for at least a year and had 1 or 2 unsuccessful TFRs. And after study start, the treatment was harmonized to nilotinib 600 milligram per day for at least 2 years, the nilotinib treatment phase. And then after confirmation of MR 4.5, treatment was discontinued. And this is the outcome here. For a second and later stop, I think that's very good. The molecular recurrence-free survival is 58% after a year. I think that's very positive. My comment to this study is, if nilotinib was given on a long period before the first discontinuation, I would not introduce 2 years of nilotinib again, because we have cumulative cardiovascular events seen in NSND, and I think a switch to another second generation drug would result in the same results. This was not done in this study, but there was not a significant number of patients with cardiovascular events. But of course, we need to observe this for a longer period. This was an interim analysis here. We need to see the final data on tolerability at a later stage. That's it. That's my summary of the most important clinical presentations at EHA. There were, of course, several others, but I need to restrict this to 20 minutes, what I did. And but we can discuss, of course, the other presentations as well. But it's mainly on the selection of the right drugs, on dosage, and of course on treatment-free remission and patient-reported outcome. I think these were the most important endpoints for all the presentations. Thank you very much.
Wonderful, thank you so much. I can imagine that it's hard to pick your highlights from the separate presentations.